You've seen this slide already today from several speakers, and we're here talking about how we could get towards ending TB in 2035. And from the modelling behind this graph here, you can see that if we implement our current tools more effectively, then we can have a substantial impact on our TB rates. When you get to the year 2025, we need to accelerate that if we're to get down to less than 10 cases per 100,000. And so the way that we can do that is to add new biomedical tools. So our current tools can do what the dotted line is here, but we need new tools, including new vaccines, to be able to reduce those rates further. So, and another reason why we need new vaccines, of course, is, as you've just heard from Dave, there's multidrug-resistant TB, and I don't know whether MDR-TB is taken into account in that modelling that we've just seen, and whether perhaps increases in MDR-TB might make it even more difficult to achieve those ends by the end of 2035. So, of course, as somebody who works in vaccine development, I'm going to argue that the best thing that we can do to prevent MDR transmission is to stop people from getting TB in the first place, and an effective way of doing that would be with a vaccine. And we do already, of course, have a vaccine for TB. So this is the BCG vaccine. This, of course, is the most widely used vaccine in the world. First used in humans in the 1920s, initially developed to prevent bovine tuberculosis, but now, of course, used to protect against human TB. So this Mycobacterium bovis was cultured in the laboratory on potato slices soaked in glycerin and ox bile, and it was passaged from one culture to another. And after about 240 times, it lost some of its genome. And of course, we didn't know that at the time. We know that retrospectively looking at it. And losing that part of the genome, the RD1 region, actually made BCG avirulent for humans and for cattle, yet still able to protect us against disease. So, and this is just to summarise, and don't worry about being able to read the individual trials, but this is a figure taken from Poonam Mantani's review of BCG efficacy. And it's just a reminder that if you look to see where these dots are lying, that in infants, BCG can actually protect against tuberculosis. Overall, the effect is about 50% against disseminated TB disease in infants. It can also protect skin test negative children against TB disease. But now, if you look at the skin test positive children, BCG is becoming less effective. And it's not effective in skin test negative adults or in skin test positive adults. So this is telling us that as somebody gets older and as they get exposed to TB or mycobacterial environment, as they become latently infected, BCG is less effective. And so today we've been hearing about this huge reservoir of latent infection, and what this is telling us is that our current vaccine can't address that problem. So we can protect people from establishing infection in the first place, and we do that by giving BCG at birth, but what BCG can't do is protect somebody who's already latently infected from developing TB disease. And this is unfortunate, and I think that was highlighted really nicely by Gwen Knight, who published this paper in PNAS in 2014. So she's a mathematical modeler, and what she did was to look at the potential impact of a new TB vaccine given in infancy versus a vaccine given in adolescence. And so here, if you give a vaccine with varying efficacy, 40%, 60%, or 80%, then you definitely can reduce the number of cases in infancy, but you have a much greater impact on ability to reduce TB burden if you give a vaccine in adolescence. And this is partly because, of course, infants are, to some extent, passive. They're the recipients of a transmitted bacteria, whereas adolescents are actively going out in the community. They've got the pulmonary form of disease they're going to be transmitting, and so you can have a much greater impact going into this population. And of course, this is exactly the population who is not protected by BCG, and looking at all the efficacy trials with BCG, if we did go in and try to immunise them with BCG, we're not going to be able to help them. So how can we improve on this? There's a nice review by Peter Anderson and Josh Woodworth who've looked at different ways in which we could improve on BCG. And one of the strategies which has been used largely so far in TB vaccine development and used by the group I used to work with in Oxford is looking at boosting immune responses to BCG. So this is taking antigens already present in BCG and using a booster vaccine, such as a subunit vaccine, a viral vector, and trying to enhance a Th1 type immune response against those antigens to try and extend protection from infancy right through to adolescence and early adulthood. So another strategy is to actually try and induce a different type of immune response than the classical Th1 response. So we know that CD4 T cells and a Th1 response are extremely important for protection against TB. We know this because of HIV, of course, and we know this because people have polymorphisms in type 1 cytokines, interferon gamma, IL-12, are very susceptible to disease. But also CD8s do play an important role in protection. We think the fairly recently identified populations of Th17 cells would be protective, and perhaps rather than these effector type Th1 cells, we want to induce more of a memory type T cell population which has got interferon gamma and IL-2. And of course, maybe we want to induce an antibody response as well. So this is a concept that we tested with the viral vector vaccine MV85A. We tried boosting a Th1 response, and we didn't see enhanced efficacy with that approach. So maybe we should look at trying to broaden out the immune response. And another way in which you can broaden the immune response is by looking at a different antigen. And so here it's looking within the MTB genome itself and saying, are there certain proteins or virulence factors which are expressed at different stages of disease, which might actually be more important for protection rather than just choosing an immunogenic protein which is expressed in all phases of disease. So it's three nice ways to think about what we can do with a TB vaccine for latency. Oops, wrong way. And one of the, certainly this is an approach which is really being supported at the moment by the Bill and Melinda Gates Foundation, is to look at boosting up an unconventional T cell population. So we know CD4 T cells are important, we know CD8 T cells are important, but actually there's a real huge and really interesting body of work coming out at the moment about unconventional T cells, and so mucosal associated invariant cells, HLE cells, CD1C. So these T cells don't respond to the classical MHC class 1-2 T cell receptors. We recognize antigen in association with an antigen-presenting cell. They tend to recognize conserved epitopes and lipid and glycolipid and sort of non-classical, non-protein antigens. And so we haven't ever really specifically focused on these in terms of vaccine development, and partly because it's difficult to know what the ligands are for these cells and how to really manipulate this with a vaccine and induce a memory response. But what we do know is that these unconventional T cells, which are quite innate in character rather than memory-like, can respond extremely rapidly upon infection. And so if you're exposed to TB, they may well get to the lung much more quickly and prevent the establishment of infection. But equally, they are likely to sort of decline at a rapid rate. We don't really understand how we can sustain them and how we can have a memory response to these cells. And so actually, going back to thinking about latent TB, if we go for an unconventional T-cell response, we might be trying to close the stable door after the horse has bolted. These people have already been exposed to TB. They've already probably got something there in their lung. So these innate cells are probably already around. And if they can't sustain a memory-type response and prevent the recurrence of infection, maybe we have to revert back to looking at our conventional T-cells, conventional memory and humoral and cell-mediated immune responses. So here are some of our conventional responses. So this is CD4 T cell, and these T cells can, we now know, differentiate into different cell types which have diverse functions. And so you get your classical Th1 type cell, which we think is important for protection from disease, a Th2 type cell, which promotes more of an antibody response and may be less important for disease, may even be pathogenic. Th17 cells very strongly Th1 type might result in immune pathology if you have too many of them, but equally are probably very effective at clearing a pathogen. And then a regulatory T cell, which is here and can actually suppress all of these other cell types and keep a balanced immune response. So this is what we're sort of reverting back to for a vaccine for latency. So what about the antigens? So I mentioned that there's a nice review saying that if we perhaps go for different antigens within the MTB genome that reflect more of the different disease states, we might have greater success with a vaccine. And here is a cartoon showing potential disease states. So somebody inhales an aerosol, you've got infection of alveolar macrophages. They might be stationary here, latent at this phase, or you might get some early granuloma and have a latent infection here. But eventually this granuloma breaks down. You get release of the mycobacteria, establishment of infection and disease, and then someone can transmit. So what are the different antigens which are expressed in these different states? Well, sadly, there's actually very small, very subtle differences in antigen expression in any of these states. So if you look where we can look, we see responses to secreted proteins, to cell wall proteins, to lipid antigens, glycolipid antigens, to those RD1 proteins, which of course are ESAT6CFB10 that we use in our IGRA diagnostics. And it might be that in certain stages we see slight upregulation, perhaps in latency of this DOS-R regulon. Although if you look at the data from human studies, you see these DOS-R proteins expressed at all phases. And you might see, upon activation of disease, some slight upregulation of some resuscitation promotion factor proteins. Again, so perhaps a pathogenicity virulence target, but you can detect the expression of these proteins at all stages. So actually, unlike malaria, the different stages of TB in terms of the bacteria actually look highly similar to each other. And so that means that the vaccine candidates that we're looking at the moment would largely work or not work at any phase of the disease in which we use them. And I think this really ties into the concept that Alison has already introduced, is in that, in fact, latent TB disease isn't a defined entity, and what you're looking at is actually a whole spectrum of response, probably related to bacterial load. And so as your bacterial load increases, you get promotion of innate immunity developing into acquired immunity, going on to active infection and then disease. And even in active disease, what you see is that the TB granuloma exists in many different states. And so the bacteria that are sitting within these different granulomas and different lesions are going to be expressing the different types of antigens that are perhaps suiting them to that environment. So even within one lung, you'll see a mixed population of mycobacteria expressing different antigens. And there's a nice study by Joanna Flynn's group where this is a non-human primate study, and they're showing what happens in response to treatment. And here's one granuloma, which is slowly resolving over time, but then you have another granuloma here, which is slowly becoming inflamed over time. And so this is within the same non-human primate, just looking at different parts of the lung. You've got lesions doing different things over time. And of course, the bacteria within those lesions, if this process is driven by the bacteria, are going to be expressing subtly different antigens as you go through time. So for me, I think, as a vaccine developer, this is really quite frustrating because, we don't have set stage-specific antigen expression within TB. So if you ask me to give you a vaccine designed for latency, I can't give you one because the bacteria isn't behaving. It's not giving me a set of antigens which I can target, particularly for latency. The only thing I can say is that this region of difference 1, which was deleted from Mycobacterium bovis during those passages and contains ESAT6 and CFP10, we know that with the BCG vaccine we are missing this, and so perhaps it won't do any harm to try and boost up our immunity to this particular protein if we want to prevent somebody who's already got a latent infection from going on to TB disease. And so with that in mind, looking at the TB vaccine pipeline, then I've pointed out here some of the candidates which contain antigens within the RD1 and so could potentially be useful as vaccines for prevention of progression of latent disease. And so MTBVAC is a whole live Mycobacterium tuberculosis, which has been attenuated to be non-pathogenic, so quite an exciting vaccine to watch. ID93 contains some of those RD1 antigens, and as does H56, which is in clinical development as well. So these, and of course, Ruti, which is also a whole mycobacterial preparation. These vaccine candidates are used for prevention of infection, post-exposure, and as therapeutic vaccine candidates. So the vaccine developers are actively looking at using them for these 3 conditions, or in Ruti, they are focusing on therapeutic. So again, and this is because we don't have those stage-specific antigen expression, and even with the RD1, giving it as a primary vaccination at birth still might enhance the ability over BCG to protect from gaining infection. So Alison showed this graph earlier on that actually, as we progress from latent TB infection, we go through a subclinical phase to active TB disease. And the things that drive us on towards active TB disease are actually host factors, not bacterial factors. And those host factors are things which impact on the immunity, and in particular impact on your T cell function and on inflammation. And so really what drives you immunologically towards active disease is not a bacterial factor, it's a host factor. And what do we know about the host in TB? Well, Anne O'Gara's group published this really nice study looking at active TB patients and healthy controls, and showed that a neutrophil-derived interferon-inducible gene signature, type 1 interferon-inducible gene signature, was highly active in active disease and came down following treatment. And so this was our first hint that in TB disease, at least, a type 1 interferon pathway might be an important thing to look at. So I've also been involved in an immune correlates analysis, and this was a study looking at blood samples that were collected during an efficacy trial of a TB vaccine. This was the MV85A vaccine efficacy trial in infants in South Africa. And there we had nearly 5,000 infants randomized to receive either MV85A or placebo. And the vaccine didn't protect. We had 39 cases in the placebo group, 32 in the MV85A group, but we put all those cases together and compared them to matched controls to see if we could see anything in the host immune response which was predictive of risk of developing TB disease. And the difference between this study and Anogara's study is Anne's study was in active TB disease. She took samples from patients, and here we're taking samples from 10-week-old infants who are in a clinical study, so they have no sign of TB disease at all. They are IgRA negative. Anybody in the household who was coughing or had sign of TB disease, the infant was excluded. So as far as we can tell, these infants haven't yet been exposed to TB at all. And what we saw in these healthy infants is that actually T-cell activation, and this is characterized by the expression of HLA-DR on the surface of T-cells, was associated with increased risk of TB disease. And the infants had about double the risk of developing TB disease over the next 3 years of life if they had higher levels of HLA-DR expression on CD4 T-cells. In this same infant population, the risk of TB disease was halved if they had a higher BCG-specific interferon gamma ELISpot response. So in this infant population, T cells, CD4 T cells, induced by BCG vaccine did seem to be associating with reduced risk of TB disease, but certainly statistically the most dominant effect was this driver of risk, which was T-cell activation. So we went on and validated this T-cell activation signature in an independent cohort. So Alison mentioned the adolescent cohort in Cape Town, and Dan Zak is publishing that paper hopefully within the next couple of weeks. And they very kindly gave me the flow cytometry data to look at. And what we saw was the same effect, where having a higher level of HLA-DR expression on T cells was associated with increased risk of developing TB disease. And in the adolescent cohort, they had time course data. In the infants, I only had one time point. And actually, the HLA-DR signature is significant at any time point up to diagnosis I look at. So if I look at all the samples or restrict it to those more than 18 months before diagnosis, or 12 months or 6 months, there's a slight increase in the odds ratio as you get closer to diagnosis, but it's not a major effect. And what Dan Zak is publishing is that in these same adolescents, then a type 1 interferon gamma signature, almost entirely overlapping with Anogara's signature, is predictive of the risk of developing TB disease. And that effect is most strikingly significant at 6 months before diagnosis. And so you're probably picking up subclinical TB disease with this gene signature. So looking at the infant transcriptomic data, which is something that I'm doing right now with Julius Muller at the Jenner Institute, what we've seen, and this is looking at the pathways level, looking at the difference between case and control infants, the dominant signature is a type 1 interferon antiviral response, upregulated in the cases versus the controls. But this is not a TB disease signature. This is not a TB infection signature. These are quantiferon negative. They have no exposure to tuberculosis as far as we could possibly measure. And yet that signature is dominant here and in cases versus controls, and predicting the risk of developing disease within the next 3 years of life. So what we do know is that these 25% of these infants had an extremely strong response to CMV, a viral infection, of course, at the time that we took their blood sample. and CMV almost significantly independently predicts risk of developing TB disease. So I think that what we're seeing in our cohort is this same interferon signature, which is associating with disease risk in adolescents, but in our case, that signature is coming from a viral cause and not TB. So just to summarize, there are very few stage-specific antigens in TB to target with a vaccine for latency, although antigens in RD1 might help. But what we do know is that T-cell activation actually prevents a vaccine from being able to work, and that type 1 interferon signaling is associated with increased risk of TB disease in both infants and as adolescents, as well as being something which is important in active disease. And in addition to that, then we know that mycobacterial exposure itself, age, and CMV are also associated with the drivers of T-cell activation. And so I think that the challenge for making a vaccine for latency is going to be looking at the host immune response and seeing if we can somehow control that or work with that, rather than being a solution in looking at an antigen for the vaccine itself. And that's it.